This video is a quick overview, if you will, of the two papers assigned for our gene duplication. The first one is a large-scale analysis and an assessment of whether or not gene duplications have occurred in the insects. I have a vested interest in the insect group, but I also thought it was a good example of how we can apply some of the concepts that we're talking about to a very large-scale analysis. So very quickly, we're just going to look at some highlights. They are looking for signs of whole genome duplication. Now, this is a very common occurrence in the history of plants, but it has not been as common, or at least there's not a lot of evidence to support it being very common in other lineages of eukaryotes. However, we do seem to have some evidence for them in insects when they look at this. Now, to do this, what they were doing is they were looking at homology versus paralogy across large scales of genes that were available in the database. And the idea is that if we get an entire genome duplicated, initially we should, of course, have every single gene that is represented twice. Now, what happens is when you have genes represented twice, many of the times that redundancy is removed by natural selection. So if you have two copies of an identical gene and it mutates, it's usually not a detrimental impact, because even if it becomes completely useless, you still have the functional version. And so after whole genome duplication, we would expect natural selection over the intervening years to remove many of the duplicates. However, there are going to be some of those genes where there might be flexibility in having two copies, or a slight variation in one of the copies might end up hitting on a advantageous position where you've got one gene doing a very specific job, maybe digesting a particular protein or doing some sort of very specific enzymatic function, and then the opposite one can do something similar but not the same, and we can get an advantage out of that. Not only that, but we might expect to see some patterns into which types of genes are lost, maybe that have very specific narrow functions and not a lot of flexibility or other close metabolic or enzymatic connections. And then other types of genes where there might be much more flexibility and more use for different slight variants of the function. And so we can use not only are all of the genes there, which we might expect them not to be after many, many years of evolution, removes some of those duplicate copies. And we can balance that with, oh, maybe we're seeing patterns across many different groups of the types of genes that are kept. And if we see those patterns, it's more likely to be a gene duplication event rather than parallel duplication events in different lineages. And so the take-home message is they do find some, not in the entire group, but in some of the subgroups of insects, and they particularly point out Lepidoptera. Where are we here? There it is. Lepidoptera and Trichoptera, which are two very closely related orders of insects. The butterflies and moths are the Lepidoptera. Trichoptera are caddisflies, which you may not be familiar with, but those two are very close related. And then the more distantly related Odonata, which are the dragonflies and damselflies. So within those groups, they did find evidence for gene duplication because they were seeing these significant patterns of similarity in the genes that were represented more than once in the genome. Now, just one last thing that I would like to talk about is I'd like to define this term because it's important and kind of relates to what we were just talking about and the methods that they used. And this, like many papers, the methods, if you get down into these methods, are probably going to make your head spin a little bit. But if you can go through the overview, maybe the introduction, and then go through the conclusion, you can begin to at least understand and comprehend most of what's going on, then you're in good shape. And of course, this outline, this guideline will help you greatly in that. Okay, so this last term that I want to define is dosage. Let me find it here. Sorry, I had it earlier. It'll come up here. There it is, dosage balance. And so dosage balance is this idea that's taken from studies of whole genome duplication in plants, and then also empirical studies in the lab where they tested what they were seeing looking at these genomes. And basically the idea is that if we duplicate a single chromosome, so if there's a non-disjunction mutation or some other mutation that ends up giving a progeny with one extra copy of a chromosome, that often is a very detrimental mutation. And we see that in animals very easily, but even in plants, that seems to not be a very common thing that can be kept, because a single extra chromosome tends to be a bad thing. However, it's much more likely that an entire genome duplication, so turning a diploid into a tetraploid, for instance, it's much more likely that those will have a less negative impact on the progeny. And so dosage balance just means that a little bit of gene duplication is often a bad thing, like a chromosome's worth of duplication. But an entire whole genome, the larger dosage of gene duplication, may have a better chance of having some sort of a survivable outcome for that progeny. So just know the definition of dosage balance. It has to do with whole genome duplication, and then this assessment of orthology and paralogy across entire genomes. And the only reason these types of studies are now possible is because we do have very large comprehensive data sets that are available for many different species. And we can evaluate them independently, pairwise. We can also evaluate them based on multiple alignments and multiple identifications. But to be able to do that, we have to somehow first have some sort of an assessment of all of these genes, whether they are orthologous or paralogous. And so this next paper gives us an overview of some of the ways that we can decide whether or not two genes are homologous. And remember, we may then need to decide, okay, are they homologous via orthology? Are they homologous via paralogy? Or maybe even xenology, right? So those are the important vocabulary words here. And I'm going to write them here. I've got a couple definitions we're going to be going here in a little bit. But just so it's clear, right? We have homology, right, which is similarity due to descent from a common ancestor. And then within that, among the genes, we have orthology, we have paralogy, and we have xenology. You should be very familiar with these already. If not, review them, look up definitions, okay? But as we are evaluating different groups of genes, we want to be able to assess, are they homologous? Okay, first, how would we do that? And then secondly, once we decide they are homologous, are they orthologous or paralogous? And xenologous is important for prokaryotic organisms. It does play a role in eukaryotic organisms. However, it's much less frequent, although there do seem to be some particular cases where it may play an important role, particularly if two organisms are living in a mutualistic or a side-by-side relationship over many, many, many millennia. So anyway, but largely we're going to focus on orthology and paralogy as we're evaluating these, particularly in the eukaryotes. So this Pearson et al. Paper is looking at a practical view of how we assess whether or not sequences are homologous. And then a little bit about, okay, are they paralogous or orthologous? And they point out at the beginning a very important thing here, that similarity sequence is not identical to homology, but it's an important tool to help us establish homology. So realize that sequences can also be similar through other evolutionary forces, primarily convergence, right? We could have maybe two sequences that have evolved into take on a similar function, and they have hit upon a very similar strategy in doing that. And so the question is, what level of similarity would we expect to see if there's some sort of convergence going on? And then what level of similarity would we expect to see if there is homology? And this can be a very tricky question to answer, because homologous sequences can be quite divergent as evolutionary pressures take two genes descended from a single common ancestral gene, but evolutionary different pressures may take them down different pathways. And so it becomes a tricky thing to do. And so here they just point out the inference of functional similarity from homology can be difficult. Functional similarity is difficult to quantify, right? Because ahead of time, we don't know how much we might expect if two genes evolving from different gene ancestors, but they're evolving to take on a same or similar function. We don't know what level of overall amino acid similarity we might expect. So to do this, we need to start with a database, a background, right? Because ahead of time we can't say, oh, I would expect X percentage of similarity if they are homologous and X percentage if they are just similar due to convergence. That's something that you can't just pull out of the air and estimate a priori. So now that we have these big, large databases, we can begin to do this. And so we're going to talk about some of the strategies that are used to do this. Okay. Now, as we do this, we need to point out some important concepts. Number one, proteins are not just amino acids. Often those amino acids are organized into structures that we call domains. Okay, and so a domain is a region of an amino acid, a region of a protein with, not a region of amino acids. It's a collection of different amino acid residues, but a large part of a protein that has a very specific function. And so in a way, we can draw an analogy between a domain and like a structure in an organism, right? So the arm of a vertebrate is a certain subset of that organism that has a very specific function. And even though it has diverged and taken on different functions, there are still some levels of similarity that we can see in the morphology of those. And so likewise, if I have a part of a protein, let's say a enzyme that has an active site where the substrate binds to an enzyme, or a transmembrane domain, then I would expect those to maintain some amount of similarity despite maybe diverging a little bit in function over years. And so a domain is really just a subcategory, an identifiable, quantifiable maybe even subcategory of an overall protein. Now, the other thing that they point out is that taxon sampling is very important. So we need to look at how many organisms we have sampled, because if we have, only a fruit fly and a human and yeast, then there might be lots of changes that have arisen since those genes in those organisms have diverged, and so much so that it's really difficult to identify homologs. But if we're able to fill in some of the gaps between those organisms, maybe we have many different vertebrates and many different insects, and then we start throwing in other invertebrates and other fungi. And if we can then fill in some of those gaps, then the evolutionary distance that we're spanning between species is going to be somewhat less. But because of extinction events, because of the evolution of major groups that have become very, very divergent, fungi versus animals versus plants, sometimes there's a limit to how much of that evolutionary distance we can span, right? Those extinct organisms for which we don't have any genome data would be great, but we just, it's irretrievable. It's been lost. Even if we find fossil remnants, we're not going to find usable genomic data. And so sometimes spanning that gap between taxa just really is not feasible. And then it also points out here that we need to make sure that we are evaluating false negatives. That's basically what this last part of this paragraph says, right? That if these methods that they're going to talk about here find a significant homology match, it's very likely, it's very highly probable that that is an accurate result. However, it might mean we might miss some of those, and so we might have many false negatives where there are actual homologs, but the processes we're using just didn't miss them because those homologs have diverged so much. So it's a complicated issue, and let's just look, we're just going to highlight some of the ways that people do this. As we mentioned earlier, the very first thing that we need to do is figure out kind of what our expected null distribution would be. Like, just by chance, how much similarity would we expect to see between two randomly selected proteins? And again, to do this, we need a large database, and luckily for us, we now have those. So we can generate those, compare proteins randomly across entire genomes of a variety of different organisms, and get an expected similarity score based on that entire distribution. And then we can say, okay, at what level are we going to now infer significance based on this distribution of similarity scores? Now, methods that use these are BLAST, FASTA, S-search. There are a couple others, but these you may or may not be familiar with. The only one we're really going to look at is BLAST, and hopefully you're already somewhat familiar with BLAST. We introduced it in some applications when we were looking at generating data sets for phylogenetic analysis. So we'll look at BLAST a little bit here. But again, the BLAST scores are going to be somewhat dependent on the overall database size. And there's a little bit of a catch-22 here where sometimes it's easier to find sequence similarity with smaller databases if they include closely related organisms. So a better strategy, rather than searching everything available, might be to say, well, let's only search among the vertebrates, for example, or maybe only among the mammals. And that might then help us to have a better chance of identifying these homologs, okay? And then another thing that they point out is it is much, much better to use a protein sequence than a DNA sequence. And I'm going to give you 2 reasons that are basically outlined here, but in a more technical terms. So there are 2 reasons why a protein alignment, when you can have it, right? You can't do that with non-coding regions, but non-coding regions tend to diverge much, much more quickly and are not really useful for these large-scale assessments of homology. So we're really looking at genes. But anyway, the 2 things that we can do when we are trying to, or the 2 advantages that DNA has, okay, let me start over. The 2 advantages that a protein search has over a DNA search are here. So I'll just list it here. Advantages of searching for protein I can't type, sorry. Homologies versus DNA homologies. Okay. You may already have some idea, so great. But here are the 2 big ones. Number 1 is DNA is much less conserved. And the reason for that is just because of the way the genetic code works. We can have many mutations in DNA that do not change the corresponding amino acid, and so we build up much more sequence divergence in DNA than we do at the amino acid level. And then number 2 is there are only 4 DNA monomers, right? Adenine, guanine, cytosine, thymine, right? The A, T, C's, and G's of the DNA. But there are 20 amino acid monomers, right? The 20 essential amino acids that build the proteins to make life. And so because of that, there's more you have a 1 in 4 chance of a random mutation causing convergence, causing a duplicate. There's just more that can occur by chance at that level of DNA than there are at the protein level, just because of the number of monomers. So for those 2 reasons, it's much, much better. In fact, always you look for homology between genes using the protein sequence and not the DNA sequence, okay? And then to wrap up, I just want to cover these 2 definitions of scores that are used to test overall similarity. The paper points out that a rule of thumb for looking for similarity is, and it's down here on this one, is a rule of thumb has been 30% sequence identity. And that's, well, over what area, and so how long of a gene you're estimating plays a role in that. So it's a very general rule of thumb. But they point out that that can be overly conservative, that there might be sequences that are actually less than 30% similar, but have very strong evidence for homology. And so they propose that a better value, rather than just overall sequence identity, are these 2 scores called an E-value and a bit score. Now, they don't define these here. They assume you know what they mean, and these are very technical statistical measurements, but you get them when you do a blast search. And so often people are using these scores to evaluate, and they're kind of cutoff points, which we won't talk about. But I do want you to have a very clear overview, at least, not a detailed mathematical or heuristic understanding of how we do an E-score or a bit score, but I do want you to have the big picture view, which is here. An E-value, which you get out of a blast search, is the number of expected hits of similar quality that you would expect to find just by chance. So let's say you take a sequence and you search it, and you get 100 different significant hits. So the E-value is, well, is that by chance, or how likely are you to find that number of significant hits just by chance? And so if an E-value is very, very small, right, going down towards 0, then basically it's a more significant result, and less likely that you're finding those results just by chance, okay? And then finally, a bit score is the required size of a sequence database in which the current match could be found just by chance. So if you find a really, really, really close match, then the idea is that you probably would need only you would need maybe a very large database, into the billions and billions of sequences to be able to find that one just by chance. But if the sequences are more similar, then the database size would be smaller. So this one relies a little bit more heavily on database size, although database size does play into the calculation here. So just look at those. If you want to write those down, you can refer to them, or just put them in your notes. You refer back to them, but you should know those two. And when you're evaluating a BLAST search, you should look at them. Okay. And then finally, the last little point that I want to make about this paper is we are really, when we're trying to do this, is trying to tease apart this relationship between function and homology. And they also point out that there has been some flexibility or some leeway in the way that researchers use the terms orthologous and paralogous. Now we're going to use the stricter definition, which is based on inheritance of gene copies from an ancestor. Okay, and so we looked at that. You had an overview. You'll have a little project number 3, that will look at a methodology for discerning orthology versus paralogy. And realize this is not always similar, because paralogous genes can have very similar function. Sometimes there can be elements of convergence that play a role, so it's not always a cut and dried decision. But in very strict terms, orthologous genes trace their ancestry back to speciation events when they broke up, and paralogous genes trace their ancestry back to gene duplication events when they became more than one copy in the genome. Now, the looseness that is sometimes used, it's probably worth being aware of, is people use the term orthologous to mean homologous genes that have a similar function, and paralogous to mean homologous genes that have diverged in function. And although that might be true much of the time, it's not 100% due to some of these convergence. And the paper points this out, that we can have paralogous genes with very, very similar. And of course, if we have a gene duplication events and then loss of some genes, that further complicates our inference of whether or not genes are orthologous or paralogous. Okay, that covers it. There's actually, the summary here does a very good job, hits many of the points that I talked about. It gives you a little bit more information, particularly about methodologies or databases that you can use to look for similarity between sequence and particularly similarity between domains, which is what the CISPROT website does. Again, probably not really relevant unless you're looking at a project or research assignment where you are trying to assess whether or not genes are similar just by chance, or parts of those genes are similar just by chance, or they were inherited from